I feel like Deborah is probably the person that needs the least introduction of everyone. I had the very fortunate 3 months of my life before the pandemic started that I shared on the same department. And I must say I enjoyed every single minute talking to her. And I'm so excited that she's going to open this workshop by giving us the ambitious talk about, what do we don't know about sex chromosomes. And I guess with this, I will let you speak, Deborah. Thank you very much.
Thank you, Camille. Thank you for inviting me. And what a big topic. But I put sex chromosomes in the middle because I think they're quite related to many other topics that will be covered. And of course, in this meeting, we may actually hear answers to many of the questions. But at any rate, I want to emphasize at the beginning that we want to recognize our ignorance, not in a bad way, but to realize that that is the route to advances. And also, I like this other Edinburgh scientist, Bill Hill. To what question is this the answer? I think that's a good question to ask ourselves regularly, trying to forget what we think we know, at least in a productive way, to be able to answer new questions. And particularly, I'm going to focus on the things that have obsessed me for a long time. Why do sex chromosomes evolve? Why are there large sex-linked regions in some organisms, but not in others? And what are the consequences of those regions being non-recombining genome regions, which leads to the very strange properties that we see for sex chromosomes compared with autosomes. And I think the word chromosome is kind of crucial here, because I want to also quote from in the abstract that I noticed Alex Sember, and this really expresses something that I've tried to say, but Alex says it briefly and clearly, and I think it's important that we need to relate cytogenetic and genomic data. Now we at last can get genomic data, and these things are mutually informative and help, I think, understand some of these questions. So I will go through some stuff, and I hope I can relate various concepts to these questions. But of course, as I said, many people will answer some of these questions, and I'm going to try and focus on things that may be a bit of a surprise or a bit of a jolt, I hope, to us to think about our ignorance as well as answers, things that we know, try to get out of our comfort zone of what we do understand. But of course, there's a lot we do understand. One of the first things is that sex chromosomes can have sex-linked regions that can encompass the whole chromosome. In the right-hand side, I show some examples of birds where one chromosome is clearly out of line in this coverage measure, showing low coverage. In females, they have female heterogameity, and the W chromosome doesn't have very many genes compared with the large number that are present on the Z chromosome. This shows a very simple analysis that can now be done simply using genome sequences. You only need one of each sex, and you can detect the chromosome whose coverage is way out of line like this. You get some surprises. Here's something which I'm calling a neosex chromosome. So sometimes two chromosomes will behave the same. It looks as if another one got hitched up, and it's also become depauperate. The W in this one has become depauperate of genes with low coverage as well. And at the opposite extreme, we have something like the mosquito. There are so many examples. I'm just showing the odd one to illustrate. This is only a very small genome region, and you couldn't detect it that way, but it has genes present. In this case, it's got male heterogameity. It has genes on both sex chromosomes, and by looking at the sequences, you can see differentiation between them, identifying the region. And Gabrielle will tell us more about identifying sex-linked regions, of course. Here's another situation in a very interesting taxonomic group of fish, the sticklebacks, which I think we're going to hear more about. The three-spine stickleback, Katie Peikel's group have done very nice work, and it's clear that one chromosome has higher diversity than the others. And we can see that that's because of differentiation, sequences that are present on the Y and not on the X. And in this other species, you see something similar. But in this thing, the tube snout, which is a very elongated stickleback, it's not obvious. So maybe something else has happened there. So we know that sometimes there's a suppressed recombination region and sometimes there isn't. And this is the first thing I want to sort of get out of our comfort zone. We tend to think of lack of recombination, meaning that recombination got suppressed because the lack of recombination is in the sex-determining region or includes the sex-determining region. We think that the presence of the sex-determining region caused suppressed recombination in that genome region. And I want to emphasize that that's not so, and I want to emphasize it by giving it a special terminology, secondary expansion, meaning that there's a sex-determining region and somehow recombination suppression happens. But in fact, I want to start by explaining that non-recombining regions can also occur as a sort of primary event. They can appear when a sex-determining locus evolves and directly causes a non-recombining region. The easiest one to think about is a duplication. If you have a turnover event so that a chromosome acquires a male determining gene, formally speaking, that defines a Y-linked region. It might be a small duplication, a single gene pops in, and that defines that that gene is Y-specific, if it's an XY system, of course. Could be W-specific in a female heterogamety case, but let's talk about a new Y. A new bit of the Y that's created by this event is Y-specific, and perhaps it will also cause a little bit that can't pair with the homologue. So because it's just not there in the homologue, and outside there may be a region of non-pairing as well. And such regions might well expand. Dan Jeffries and colleagues have proposed a neutral model in which this lack of homology could actually lead to that region expanding. So that would now be a secondary expansion, and it would be indeed because there's a hemizygous region there. So it's a different kind of suppression from what we often think of, where it's often considered that we have interacting genes in a Y-linked region which select directly for lack of recombination. This would be quite a different route to producing a non-recombining region. And it's not very implausible. Turnovers occur in many organisms. This is just taken from these data from a review, one of the reviews Dan mentioned, of particular taxa in fish. Turnovers have been discovered many times, and the genes have been identified in some cases. Sometimes the turnover occurs because a new male determiner or female determiner has occurred by what they called in the review diversification, a mutation in one of the genes, standard genes that are often involved in sex determination in other critters. But sometimes the same gene even sometimes is involved in duplication events. And in fact, in this compilation, which could be out of date now, shows how fast things go. 2021 could well be out of date from what the figure that Dan just showed. There are more duplication events than diversification events. So it's common, but a duplication event can actually cause, or a new male determiner arising by a diversification event could also cause a large Y-linked region, because if it occurred in a region which doesn't recombine, it will produce a large region that doesn't recombine. And we stumbled across this in our work on the guppies, where we found that in the males, the genetic map looks like this. This is the physical position. Now we can do genome assemblies and understand the relationship of the genetic map to the physical map. We can see that recombination in females increases, the map position increases as you go along the physical positions in the chromosome, but in males it doesn't until you get to the very tip. This is called crossover localization, and it's very strong in the guppy. And so if you had a turnover event on a chromosome where a male determiner appeared by diversification or duplication, if it occurred, say, in one of those middle bits that doesn't recombine, it might produce a large region that at least doesn't recombine very often. Maybe there's occasional recombination, there's evidence there is in the guppy, but all the same, it would produce a large region that would behave like sex-linked much of the time. And we know that this, in the guppy, this is probably something that occurs on all chromosomes, this pattern, based on analysis of GC content, because GC tends to be high when you have a lot of recombination. And we see a pattern like the one I've plotted on the right. That's for the same chromosome as the one on the left with the genetic map, but the other chromosomes look the same. And when you look at outgroup species, it looks very much the same as well. So this could well be the ancestral state, where much of the chromosome has low recombination or maybe zero in males. And then you have this possibility of creating a large Y-linked region in a single event, in a primary sex determination. Oh, wrong way, sorry. And crossover localization in both sexes would be exactly the same in terms of if you had a male determiner appear on a chromosome, it would create a large region that doesn't recombine very often. This is an example from a plant, from Stephen Wright's lab, from Joanna Rifkin, where they showed that in a Rumex species, the XY pair at the right, and all the chromosomes have this property, and the XY pair does seem to be a large region that's just like the other chromosomes in this respect. So it's an example where probably this process has happened. So you could create large, potentially large Y-linked regions, maybe not completely Y-linked, by this kind of primary mechanism, or you could create it by secondary selection to suppress recombination. And we need to understand what organisms probably do have large and which ones have small regions of sex linkage, and try and figure out which of these mechanisms, and there may be other mechanisms that I haven't thought of, is involved. So we know, of course, that evolutionary strata are often found on sex chromosomes, and I want to emphasize that that seems to me to tell us that secondary suppression of recombination has happened. Parts of that chromosome were not non-recombining in the past, and now they are. The strata in mammalian sex chromosomes, originally discovered by Lahn and Page in human X and Y comparisons, and then studied by Melissa Sayers and Katerina Makova in detail, that seems to show that this is what's happening. And we can see that the oldest stratum, which was sex-linked in marsupials, has very high divergence between the few genes that are left, whereas the younger strata have more genes compared with the original numbers. So these are the original numbers of ancestral genes that Melissa and Katerina established by using outgroups, make sure that they were really old genes that had been on the XY pair. And these are the divergence, these dots plot the divergence. You can see high divergence going to lower divergence as you get closer to the pseudo-autosomal region that still recombines. And that's a sort of paradigm. And again, I want to shock people a bit, although everybody may know this already, but I was shocked to discover how many species don't show strata, or how few species are known to show strata. We know eutherian mammals, it's very well documented. Three-spine sticklebacks, schistosomes, Beatrice vicosa showed using degeneration that there are degenerated and less degenerated strata. Birds are complicated. Their ZW chromosomes are not yet as well understood as mammal ones, mammal XY. Maybe snakes and some plants perhaps show strata, but again, a database might be able to help us learn more, focus on the species that can help us learn more. We don't know the mechanism either. There could be a changed crossover pattern, something again we tend to ignore, maybe a changed pseudo-autosomal boundary, loss of a hotspot, we don't know. Our work in the guppy suggests it's not loss of or not change of a pseudo-autosomal boundary. We can't say whether it's inversions because we don't have a good enough assembly yet to be sure. But in some organisms, inversions do seem to be important. In papaya, the assembly of the very small bit, just these little bits that are sex-linked out of the chromosome in question, out of the sex chromosome, you might call it, those small bits do seem to be inverted in the Y, or between the X and Y, there's inversions, a couple of inversions. And the inverted bit corresponds to a stratum with high divergence. This is synonymous site divergence. And then going down as you go to the recombining bit towards low divergence. So this may well be an inversion that was selected perhaps for lack of recombination, a secondary suppression of recombination. A better example still is the stickleback that I already mentioned, and we may hear more about it, but here there's definitely different strata. An old one, which you can see doesn't have that many genes, quite a lot of genes are lost, with high divergence, and then younger regions with a much higher gene density versus the physical map on the x-axis, and much lower XY divergence. And in the assembly from Katie Peikel's group, there are indeed inversions. So that's nice. But there's a little caveat. Inversions are involved, but I think this is my attempt to understand what might have gone on, and Katie and her people may have much better analyses. This is just me doing it for fun at weekends without any software or intelligent involvement. But it looks to me as if some of these inversions that have happened must have happened after the recombination stopped. So that's a caveat. Just because we find inversions, it doesn't mean that the inversions actually created the sex linkage in this secondary kind of suppression process. But some of them in the stickleback, I'm sure, must have, unless I've got it wrong. You can see it's a bit complicated, and I'd be happy to be told I'm wrong. But I think that some of them probably must have been involved in creating Y linkage. So that's a nice example. We've got the humans, we've got the stickleback, and we don't have very much more. The humans, two of the most recent strata, the small one, were shown to be to involve inversions by Gabriel Mari's group. So, all right, we know that sometimes secondary recombination suppression happens. And for a long time, we discussed one model, which was selection for interacting genes that a gene that interacted with the sex-determining gene so that it was better to have one allele in males and the other allele in females, sexually antagonistic selection, like it's called. And that could indeed suppress or select for suppression of recombination. But of course, I'm sure everybody here knows, sorry, telephone. Brian will look at it. Can you answer, Brian? Can you answer the phone? Sorry, Brian didn't hear the phone. So we know that other ideas have been recently proposed. I already mentioned the one by Dan and his colleagues, and Paul Jay, who's also involved in this workshop, and his colleagues, has proposed an idea of lucky inversions that might be able to spread and fix on the Y chromosome only if they're linked to, if they're in a genome region linked to a male determinant. When an inversion occurs on an autosome, they're predicted to be eliminated unless selection favors them. But in this model, which many of you may be familiar with already, inversions should be eliminated more slowly. We're not yet clear, I think, quite how much more slowly. So some of them might not get eliminated, and they might be lucky enough to get fixed in the Y chromosome population. And allow a bit to become fully sex-linked. Colin Olito has modeled this and shown that small inversions are least likely, I've put the opposite of what I meant here, least likely, or most likely, so least unlikely to establish. So they're most likely to establish. So you'd expect, like in Paul's diagram, a succession of small inversions gradually turning the pseudo-autosomal recombining part of a chromosome into a non-recombining part. And Lenormand and Rose have a similar model in which there's a different selective situation that might allow this lucky inversion process to happen. And it could be a gradual succession of small inversions, or should probably be based on Colin's modeling results. So we can ask, do divergence patterns suggest discontinuous strata as you would have if you had big inversions, or like in the stickleback, or lots of small inversions that you couldn't distinguish from a continuous loss of recombination? In Silene latifolia, this is a very old figure, but I put it in to show that in that study, it looked as if there is no sign of discontinuity. But this is a big genome. It's currently being assembled by several groups, and more sequences are needed for proper testing. So we probably can expect advances in this. But selection to maintain adaptive alleles is still possible. When we see discontinuous large strata, it might even be indicated. Now to try and talk a little bit about the consequences of absence of recombination. I would guess, I'm guessing, and again, much more comparative data are needed, but I think the first consequence is probably TE accumulation, transposable element accumulation. Beautiful work by Doris Bactrog and colleagues showed this in Drosophila miranda, which is a young neosex chromosome, a piece of autosome that became sex-linked, as I'll describe shortly. It has very low divergence from the between the newly sex-linked sequences. So it occurred quite recently. And we see massive accumulation of transposable elements. The Neo-X, which was an autosome till recently, has a sort of normal autosomal level of repetitive sequences, and the Neo-Y has lots more. This just shows you one example of a gene, what it looks like below in the Neo-Y and what it looks like above in the, or of a region in the Neo-X. A dramatic increase in the amounts of repetitive sequences specifically identified by transposable element insertions. And we know that this kind of thing can happen quickly. We know it not just in sex chromosomes, but for example, this paper in Rice shows a sudden, inexplicable increase in repetitive sequences. So if a male determining region stopped recombining recently, we expect sequences to accumulate. They would accumulate probably quite fast, and even it wouldn't require complete suppression of recombination. Even a region which became rarely recombining might show this effect. It may be difficult to analyze these things because if they're found all over the genome, especially in a large genome, it might be difficult to actually map them, and it might be they might be look as if they're all over the genome, and I think there are interesting problems to be solved in doing that kind of analysis. Here's another example just to illustrate how quickly these things can happen. These two Gambusia species were thought to be two races of the same species. It turns out they're not. In Gambusia holbrooki, it has one chromosome is the sex chromosome. It's a different non-homologous chromosome in Gambusia affinis. And in Gambusia affinis, the W chromosome, it's got female heterogamety, is a giant W. And you can see now in quite recent studies, this one is just one of 2 recent studies where the Z chromosome has been physically assembled and the W chromosome on the right has been physically assembled, and you can see lots of gaps and they're full of transposable elements. So this is quite recent. I'm not sure how diverged other sequences are between these 2 species, but they're not that diverged. And giant chromosomes are probably caused by this kind of expansion in all kinds of organisms. Suzanne may talk about her work on the Coccinea grandis Y, which is another giant Y chromosome in this case in an XY system, and other related species don't have that. And here's a frog species with a giant W chromosome. So it happens in Z, W, and XY systems, and it can happen fast. So I guess it's the first consequence of becoming non-recombining. Of course, you also have, like in humans and other organisms, the sex chromosome, one sex chromosome is often small. And in birds, W chromosomes are often smaller than the Z chromosomes. This is a very nice study showing lots and lots of birds. And this may also reflect transposable element properties. It could reflect deletion of repeat-rich gene-poor regions once genes have been lost, as I showed you in the human older strata and the stickleback older strata. Then transposable elements could cause chromosome rearrangements that cause loss of chunks of the chromosome, and if there are very few genes or no essential genes in them, they can be lost. So you can explain conveniently both increase in size of things like W and Y chromosomes or decrease in size. You can explain everything. Okay, so that's all straightforward, and it's stuff that we can get from cytogenetics as well as genome sequencing, and we can hope to reconcile the two and understand by genome sequencing more of what we see in the cytogenetic studies. There's one limitation of cytogenetic studies, which I think is worth mentioning, is that heteromorphism doesn't necessarily mean genetic degeneration. If you get a small Y chromosome, it doesn't mean it's a highly degenerated Y necessarily. There can be other chromosome rearrangements. And in particular, neosex chromosomes create heteromorphism in a completely different way. If you had a chromosome attached, like in the Drosophila miranda case, to a sex chromosome, it would create cytogenetically recognizable heteromorphism because the 2 sexes would have different karyotypes. It doesn't mean, though, necessarily that the Y chromosome, if it's an XY system, stops recombining. If you had a small sex-linked region, like some of the ones I've mentioned, like the mosquito one, then everything could continue to recombine, provided that both sexes actually undergo pairing and recombination. And that's true even if we had a system where one sex, well, a system like this with a whole chromosome arm non-recombining, like a Y chromosome. Unless you have a situation where one sex doesn't recombine, a fusion like this, attaching something to a sex chromosome, doesn't necessarily stop recombination. As I've drawn in the bottom right, I've drawn the recombination symbols XXX. Absent, that would be true if that one sex, like males in this case, doesn't have recombination. But if the males do have recombination, then the autosomal arm that gets stuck on could still carry on recombining. Maybe it'll stop a little bit near the breakpoint because maybe they can't actually get together and pair, but it could still continue recombining, as we see in some organisms. In Drosophila miranda that I talked about, it's a case where Drosophila males don't recombine, so it does stop recombination across the whole chromosome, but that's not necessarily the case. Oops. So degeneration and TE accumulation will occur when recombination stops, or when it is largely stopped in the case of TE accumulation, but they're not the same thing. Even when we have a high accumulation of TEs and heterochromatinization, we may not have anything that we would call degeneration. That's a separate phenomenon. And we know that heterochromatinization may not even have a detectable effect on fitness. You can delete the heterochromatin without affecting fitness. So it's a very interesting question that I think more information that may come out of this database will help us is to ask how long it takes before degeneration becomes detectable. To do that, we need to decide how to measure degeneration. We can, as I said, look at read depth, we can look at coverage, and we can see that it's low in one sex or the other, which does suggest that we've got degeneration, but it doesn't estimate it quantitatively or how much. And we don't know whether TE accumulation might affect gene expression and contribute to degeneration. We also don't know whether active processes are important. For example, if TE accumulation does damage gene expression and makes it non-optimal, maybe we would select for things like an increased gene copy number on sex chromosomes to try and keep up the proper expression level. So there's all kinds of things, but we do know that degeneration can occur in terms of actual loss of genes. The numbers of genes remaining out of the number of ancestral genes that Melissa and Katerina determined is very low, very high proportion lost in the oldest strata of mammals. And it's still low on the youngest strata, but it's not as low. And then numbers of pseudogenes is another way you can actually try and estimate the extent of degeneration. But I think we need information on both kinds of measures. And at the moment, we often, if you try to look at the relationship between synonymous site divergence, for example, and degeneration, some species have been studied in terms of the number of genes still present or the proportion. Some have been studied in terms of the proportion that are pseudogenes, and not many species have been studied with both measures. And it's a kind of difficult to put together. What you'd like would be a database of such information. In Drosophila pseudo-obscura, we know that the neo-Y stopped recombining probably about 15 million years ago based on divergence of other sequences between other members of the obscura group. And this time was enough that basically complete degeneration has occurred. The pseudo-obscura Y, it got fused to a pre-existing Y chromosome and degeneration, or sex chromosome, and degeneration is complete since that time. So we can't estimate divergence between X and Y because there's nothing left on the Y. But of course, that's only telling us that was enough time. It doesn't tell us how much time it actually took. Complete degeneration might have occurred in a fraction of that time. Degeneration, as I showed you, is much less in, no, I didn't show you yet, is much less in Drosophila miranda. So we know that the time of divergence for Drosophila miranda, which is a couple of million years, is not enough time, at least in that case, for complete degeneration. We need lots more species, though, with degeneration, intermediate levels of degeneration, so we can try and see if there's any overall, even rough relationship between the amount of time or the amount of synonymous site divergence, which measures generation times, that's what we really want, and the level of divergence by some of these measures. We do have something good from the stickleback again. I keep referring to it, and I hope others won't be cross with me. As I showed you, degeneration in the oldest stratum, which is a lot less than 45% degenerated, and it's not complete. So we have an intermediate between the two Drosophila species, and it's intermediate in degeneration. So that's interesting. And we need many more species and studies of this kind. These ones are pioneering and show what can be done. Doris Bactrog has modeled things theoretically, but we can't yet test, I think. We need more studies to test whether what's predicted is actually what's observed or something like it. We can detect ongoing degeneration, which I think I'll pass over quickly, from reduced variability, because very old and very degenerated strata should not show any effects. They should have finished doing anything, whereas younger strata that are still degenerating, the processes that are modeled in Doris's paper, I haven't time to go into details, will reduce diversity. So you can see if it's still degenerating by asking whether the Y chromosome does have diversity reduced more than you'd expect from the different numbers of X versus Y chromosomes. And that's what you see in Silene latifolia, and Amanda Laraquenti and Andy Clark showed it in Drosophila pseudo-obscura. So we could at least ask from diversity studies, which are a lot of work, whether we've got an old stratum, whether a particular stratum that we think we have is still degenerating or it's not, which could also be useful in helping to get time course information. I'm not going to talk about adaptive silencing, except I put a slide in to say that this is an important question, whether it happens as well, and people have developed theoretical models, Alan Orr and Yusuf Kim, and no, not Yusuf Kim, well, maybe it is, sorry, senior moment, and people have developed approaches for studying it, Stephen Wright's group in particular. I pass on because I'm running out of time. Of course, once we understand the time course of degeneration, we want to relate it to the time course of dosage compensation. How long does this take to evolve? This is often defined rather misleadingly, like in Wikipedia, as if it is a process by which organisms equalize expression of genes between the two sexes. That's true, it has that effect, but this is misleading because it conveys the idea that that's why dosage compensation evolves. Where, for instance, this review and many others by other people, I just cite this one, it's funny, they only cite animals. Dosage compensation is better explained as making sure that the balance of genes is whether the expression of genes is correct in the heterogametic sex. That will be selected for because you've got imbalanced transcripts, but it's also selected for because you want to have the optimal level of expression in both sexes, which presumably was there before this chromosome evolved to be a sex chromosome. So we have to be careful about mechanisms that may select for dosage compensation, but I mostly want to emphasize the time course. In our work on the guppy and its outgroup species, we and Judith Manck's group came also to exactly the same conclusion. We found that the closest outgroups, these species I call micropicilia, have completely degenerated Y chromosomes. That's why I drew dotted lines here, whereas the guppy has non-degenerated Y chromosomes. This is just showing the data, mean count representing coverage in females and plotted again in males plotted against females. For autosomes, you see equal coverage of the different sequences in the experiment, but for the sex chromosome, you don't. You see half the coverage, like I showed you earlier for the Lepidoptera. This is the same homologous chromosome, and we know that an outgroup species, the platyfish, the sex chromosome is a different chromosome. So we have this kind of a phylogeny, and it is thought by Manck's group, this is a paper from their group, which I think is entirely plausibly suggesting that it's in this common branch common to the Micropocilia species that this degeneration happened. Dosage compensation also evolved. They showed that, Manck's group, in very beautiful work. So in this apparently roughly 3.7 million years, it's a bit longer than the Drosophila miranda neo sex chromosome had, but which is very little degenerated, little longer. This Y is apparently completely degenerated. That's an interesting conclusion. Of course, it's possible that it actually evolved earlier and that there has been a turnover in the guppy or even in both the platyfish and guppy lineages. So it's not conclusive about the timing, but it's the kind of data that I think we need. I've mentioned very, very briefly in passing sexually antagonistic selection, and I just want to point out that dosage compensation, evolving dosage compensation involves sexual antagonism, because if males have lower than optimal expression because of degeneration, higher expression will be favored in males, and expression going up in males, like has happened in Drosophila, might actually not be a good thing for females in some for some genes. If expression is controlled in a sex-specific way, of course, that wouldn't be a problem. Males would get what they liked, a higher expression, and females would not. But that would have to already exist, sex-specific expression, which might or might not pre-exist. And of course, we should remember that some genes, actually a lot of genes maybe, we don't quite know yet how many, have different expression levels in the two sexes, and sometimes low expression in males might be favored. So dosage compensation would not be selected for such a gene. It would actually be advantageous. And there are other selective situations, increasing dosage compensation, evolving dosage compensation by increasing the activity in X-linked genes might lead to the further evolution of dosage compensation by downregulating expression in females, as we see in mammals. So there's a lot of complications, and I have very little time to cover them. But I want to point out that Lenormand and Rose have recently proposed a model I mentioned earlier, where dosage compensation evolves over the same time course as degeneration. And in fact, that was the same idea vaguely, a similar idea, was already proposed quite a long time back by Deborah Charlesworth. And I think these ideas are maybe worth revisiting, because I've talked about the time course as if degeneration happens first and dosage compensation happens, but we could be wrong about that. These ideas are challenging that, and now we're in a position to get much more data about the time courses. I think the two things may illuminate one another in a very helpful way. Now I want to change gear totally, not really totally, and talk about haploid plants, and maybe Suzanne will talk about them again too. But again, I think we have a situation where we really, really do need an improved database. In haploid organisms, we have to talk about dioicous species, not dioecious species, and forgive me if I get it wrong, because I'm old and we used not to do that. And we have to talk about U and V chromosomes, not X and Y. Again, this is a change in my long lifetime, but I'll try and get it right. The question is, is this a very ancient sex-determining system that goes back right back to when bryophytes evolved long, long ago? Is it a homologous UV sex chromosome? It's not easy to tell. You could ask whether the UV sex chromosomes of different bryophytes carried the same sex-determining genes, but we have to be a little careful because sex-determining genes can move around, and turnovers, as I've mentioned a few times already, turnovers can happen so that a different chromosome can be the sex chromosome, but if it carries the same sex-determining genes, it might still be a different chromosome, but it might be because those genes have moved. So I don't know whether pseudo-autosomal regions are found on bryophyte sex chromosomes. Genetic maps suggested in one of the species, Ceratodon purpureus, Stuart McDaniel's genetic mapping showed pseudo-autosomal regions. They're not so clear in the genomic data, and I think more genetic mapping work is probably going to be useful. A problem with these haploid plants and asking these kinds of questions is that polyploidy seems to happen. And of course, once you've duplicated your chromosomes, it's interesting, very interesting to ask what happens to the sex chromosomes. Can you maintain genetic sex determination? What happens? But some bryophyte species have definitely lost the dioicous condition of having separate male and female haploid gametophytes. They've become what is called monoicous. How that evolves, is it something to do with polyploidy? Is it something special? Are these all derived from pre-existing sex chromosome dioicous species? Many interesting questions. And can a monoicous species re-evolve dioicy? Does it resuscitate? Do we have a resurrection of an old VW system, or does it re-evolve dioically by having a new system? And do the new sex chromosomes start to take on the characteristics of old ones, as they do in turnovers like Drosophila miranda, or fusions? We also have lovely predictions about degeneration in haploid plants. U and V should degenerate equally because there is no counterpart of the vegetative, as we might call it, diploid stage that we have in diploid organisms. Neither sex, the vegetative stage is haploid for both sexes. Bull predicted that degeneration was probably going to be minor because the chromosome would carry all sorts of things important in the haploid stage. So neither the U nor the V could lose those things. So only things which were male or female gametophyte specific could get lost from either one sex chromosome or the other, but the vegetative stage, important functions and genes for them would have to be maintained. Only sporophyte specific genes could be lost, but the sporophyte is small and parasitic on the dominant haploid stage. Actually, what we are starting to see, I think, from studies of haploid plants is that degeneration is major. The Marcantia U and V do seem to be small and highly degenerated, but that may be because of the large amount of time that has been involved. And we would like to look at younger sex chromosomes. This is a younger one, probably, or at least partly younger. The Ceratodon purpureus, Stuart McDaniel's group studied, and the genome was assembled and analyzed by Sarah Carey. Very nice work. You can see the autosomes are kind of respectable chromosomes with genes at much the same density on each chromosome, but the U and V are very strange. They have taken on the characteristics of the other bryophyte sex chromosomes with very low gene density. The low gene density, though, is partly due to very high repetitive density. TEs are very high compared with the autosomes, as you can see. But there are still plenty of genes. They analyzed 402, I believe, pairs that are present on both the U and the V. So degeneration here is not complete, and probably large parts of this U and V only acquired these genome regions and these genes and started diverging quite recently. So there's a lot more to be understood about this system, but I think other bryophytes may also illuminate this system. So then I want to end with, of course, I could have many slides about other puzzles and things we still don't understand. I've just given you a sample of things we don't understand. Turnovers, so that we go, oh dear, I've gone the wrong way, between X, Y, and ZW, or the reverse. I should have put arrows on both ends. These are not well understood. They're documented. We need probably a database that documents more of them. But we need to get into detail and try to understand why. Creating new sex chromosomes out of B chromosomes or out of nothing, perhaps, that's another possibility that seems to happen in some fish and maybe other organisms, and will be interesting to study. I think it's interesting to ask questions about XO systems that have no Y, or WO that have no Z chromosomes in female heterogamety. The complete degeneration of a Y chromosome can even go so far as to mean that there's not even any male determining gene left, and sex determination is done differently by a balance in the counts of X chromosomes and autosomes, or W and autosome counts. We know several organisms where that's probably happened, and including those micropecilia that I mentioned as close relatives of the guppy. It doesn't seem as if there's any gene left on them, not even a sex-determining gene. So one would like to understand how that can happen. Of course, to lose the Y, you've got to lose not only the sex, the male determiner, you've got to have a new system evolve for male determination, but you've also got to lose any genes which are important for male function. That's, or you've got to reacquire them on the Y, but you can't lose that. Another puzzle which I've mentioned in passing, giant sex chromosomes, but I mentioned giant Y or W chromosomes. I didn't mention this horrible case, which has just been published, of a giant X and Y pair. Well, of course, I have mentioned fusions and things expanding the size of a Y chromosome, and then it degenerates maybe. But in this particular beetle species, it isn't obvious that a lot of chromosomes have been added onto the X to account for this huge size. Of course, you can have transposable elements expand the Y, but why would you have an expansion of the X as well? Well, I've just said, again, as a cop-out, we should study more beetles, especially because beetles are really at the beginning of understanding sex chromosome genetics and evolution, as is the guppy. But I think it's a group that could be very interesting. So I end up with a wish list, if I've still got time, tell me if I've got to shut up, that I would have loved to see in a tree of sex, and I hope to live to see a new tree of sex that has much more about chromosome morphology than the old one. Heteromorphism and homomorphism, I would like to see, but I would like to see neosex chromosomes distinguished from heteromorphism due to expansion of repetitive sequences. I think it would be really, really nice to have clearer indications of outgroups. Ideally, of course, with divergence times, it would be lovely to be able to compare synonymous site divergence between a species and its outgroups with divergence between X and Y or WZ gene sequences to understand how the strata evolved in relation to the times when the species evolved from their outgroups. Of course, a separate thing which I couldn't expect to be in the database, but would be ideally related to it, is information on whether genetic maps are available. And ideally, those should be, of course, between separate males and females, so that we can see whether there are large non-recombining regions, perhaps in both sexes, that could explain why there are large sex-linked regions, even though a sex determination system is recently evolved. I think it would be really nice to know centromere locations now that genome sequences are allowing us to see that, and repetitive sequence densities can sometimes point the finger pretty clearly to where those are, sometimes not perhaps, but where it's known. Genetic maps can also tell us where the centromeres are, because if we have the non-recombining, we have a large non-recombining region, as I've mentioned, that will tell us that the centromere is located in that non-recombining region, especially if it's very big. That can be helpful. It may not pinpoint it exactly, but because the whole region may have high repetitive sequence densities, but it sure is helpful. I would like to just end by saying that taxa with conserved chromosome arms can be very useful, like the Muller elements in Diptera, where we can recognize the same chromosome in distantly related species. In nematodes, there is a similar system named after the scientist Nigon. I'm not sure about liverworts. The problems of polyploidy seem to me a bit of a headache, but liverworts may be another. And there may be many other taxa where we could do this. And again, that would be lovely to link to any database. And I'll end with a grouch. I'll end with saying thank you for listening to me for all this time, if you haven't gone to sleep. But let me mention a couple of disappointments with the Tree of Sex database. When I went to the plant one, all I got was this error message. This is probably my fault because I'm very old and things never work for me. So I won't condemn it, but I hope it'll work better next time. And a grouch about this genome sequencing. I looked to see it because I thought this is wonderful, sequencing all these organisms. We'll have the British dioecious plants like the stinging nettle. And I looked, and they have it. They have its genome size. It's a bit daunting. It's big, if you can see down at the bottom right, a gigabase or so. But all the individuals they've sequenced are males. How about a female? So thank you, and I'll shut up.
Awesome. Thank you so much, Deborah. We've got a lot to do to answer all these questions. So we've got 5 minutes, I think, for questions. So if you just put your hand up in the room and we'll give a microphone to people if you have a question. On Zoom, if you raise your hand on Zoom, we've got Paul on the laptop who will just wave and we'll unmute you, or you can unmute yourself actually, I think, on Zoom. So any questions for Deborah or comments? Or yeah, Jim, go for it. Do you have a, just grab a mic if you've got one, yeah.
Hi, this is Jim Human. I'm curious what your thoughts are on why the UV sex chromosomes in bryophytes develop size asymmetries. You would expect by default the selective environment would be symmetric for them, but they end up being very different in size sometimes. Yeah,
well, you were awake at least for my first slide where I showed an example. I'll say something completely speculative. I would guess that there have been turnover events in which a dioicous species has become monoicous. And you had gametophytes that had both male and female functions, and the sex chromosomes didn't know what to do, putting it briefly. And something strange happened where genes that were important for the gametophyte stage perhaps migrated onto chromosomes. One of the things I didn't mention, which are emerging from genome sequences, is that genes can move between sex chromosomes. There's been work for many years in all kinds of organisms. Genes can move off the sex chromosomes and also onto sex chromosomes, as all the Drosophila Y-linked genes, for example, are not genes with homologs on the X. They've moved on to the Y. And so if something like that happened in the bryophytes and genes moved off the sex chromosomes, you could end up with one of them vanishing and the other one perhaps for some other reason because it's got genes with functions that are different being maintained. And then if you re-evolved dioiche, maybe if it was recent enough, you could still see that size difference, but that's entire speculation.
Do we have one on Zoom? Or, yeah. Raise a hand. Oh, yeah. Okay, Ji, if you want to unmute yourself, you should be able to ask a question.
Yes. Can you hear me?
Yes, hi.
Hi, Deborah. I'm sorry, my camera is not working.
Yeah.
I'm very interested at the, so you mentioned about the T accumulation of the recombination is suppressed. So there are, in some species, the scenario could actually be reversed, like the inversion case, that one of the autosome could actually start to accumulate, repeat before it turns into a sex chromosome. We didn't really have such case, but in some species like platypus and some reptiles, for unknown reason, one autosome just become giant and also misleading, you know, like many people who study typology could recognize it as a sex chromosome, but later it was found actually a pair of autosomes with very different sizes. So I wonder if you have any, you know, comment on such pattern that one autosome of a pair could just start to suddenly accumulate TE, and it of course raises its chance to become a sex chromosome.
Yeah, well, I'm not sure of a really clear case, but you've mentioned some, and I should go and reread some of those papers. There's the one I'm aware of is in Triturus, the newt, which has what I believe is an autosome, which is extremely heteromorphic. That can sometimes happen. My understanding is that extreme accumulation of repetitive sequences is usually a sign that the genome region has become non-recombining first, that the accumulation is a consequence of being non-recombining. So, and I'm not sure, I don't think you were saying different, I'm just saying I think that's probably the case. So there are different reasons why autosomes can evolve non-recombining regions. Supergenes, for example, are known in some organisms, and in Triturus, I think they think there's some kind of a supergene, but I don't think it's yet fully understood. If Ben Wielstra is here, he may be able to answer it. So I think that's true, and I think these large non-recombining regions that I illustrated and rather emphasized, maybe overemphasized, those would be regions that would accumulate, and the genomics shows they do accumulate a lot of transposable elements, but they're not dimorphic in the same sense. So I think what I said in the talk, that if a male determining region occurred by a new male determining factor popping up in such a region, then you could end up with one haplotype of that non-recombining region being confined to males. Then it would define a Y-linked region. It would become confined to males in its transmission, at least if there's no recombination, and it would acquire new male determining insertions and of transposable elements, new male specific insertions of transposable elements, and new male specific mutations, and it would gradually diverge. That's true, but I don't think there's any other reason why it would predispose to becoming a sex-linked region or sex chromosome. I think it is a possibility, of course, but I think it's a subcategory of that possibility that I already raised, where you have a large region, for whatever reason, that's non-recombining, although it's ancestrally an autosome, but it's definitely one of the possibilities that one should bear in mind. Yeah.
Okay, so I think we are scheduled now for the in-person attendees for a coffee break.